This is the video for Lab 2 on the scientific method. Science is one way of looking at the world, and to get scientific information, you have to be able to observe and experiment on the process you are looking at. Another thing about science is that it strives to be objective, that you try to avoid personal bias in determining what the facts are. And the last thing about science is that it constantly changes. As more people make observations or perform experiments, we come to new conclusions, and so the idea of the scientific information can change in response to new data. The scientific method is the process by which we determine scientific information about the natural world. And the scientific method has 5 different stages. The first is observation, then question, then hypothesis, experiment, and finally conclusion. The very first step of the scientific method is to make observations about the process that you are interested in. And direct observation is where you are directly observing that process. But you can also read about what others have observed and discuss with others either what they have directly observed or what they have read. But the very first step of the scientific method is gathering information either from direct observation or from others what they have observed or what they have read. Once you have made a lot of observations, either directly or discussing and reading what others have observed, now it's time to form a question. And when you are forming a scientific question, it needs to be well-defined and specific. The more specific the question is, the more likely you will be able to get scientific data on it. The other thing about the question is that it needs to be measurable. So you need to be able to measure some sort of result, like a change in length, a change in height, a change in speed. So a good scientific question is going to be well-defined, highly specific, and measurable. If you ask somebody, what is the meaning of life? That is not a good scientific question. It's not well-defined, and it's definitely not measurable. But if your question instead is, does vitamin D decrease the symptoms of depression? That is very well defined, it's specific, and it is measurable. The next stage of the scientific method is hypothesis. And a hypothesis is often referred to as an educated guess. And it's a guess as to the possible answer to the question. The reason it is referred to as an educated guess is because you've done all the observation ahead of time. So a hypothesis is a pretty good guess as to what the answer to the question is. And a hypothesis, like your question, should be specific and it should be measurable. And often a hypothesis is written in an if-then format. So the statement, the sentence would start with if something, 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 then something, something, the result. So for example, if you have done a lot of research on drinking milk and bone development in children, a possible hypothesis could be, if a child drinks 3 glasses of milk a day for a year, then the child's bone development will be normal. Once you have made your observations, formed your question, generated a hypothesis to that question, now it's time to experiment to determine if your hypothesis is correct or not. And the purpose of the experiment is to generate data. So this is the information that is going to support or refute your hypothesis. And again, data is usually information that has been measured, like a change in weight, a change in speed, a change in height, and so on. When you're designing your experiment, there are different variables you have to keep in mind. Also, you will divide the subjects into control and experimental groups. And then repetition is very important. So the more times you repeat the experiment or the more individuals you test during the experiment, the more reliable your data will be. When you are designing your experiment, there are 3 different types of variables that you need to know. And these types of variables are very important in this class because in every lab and almost on every quiz and on your lab practicals, you will be tested as to whether you can identify the different types of variables. The first type of variable that's involved when you design an experiment is the independent variable. In a well-designed experiment, there will be only one independent variable, and that is the variable that you are manipulating, that you are setting up at the beginning of the experiment. So this is the variable that you set up at the beginning. The second type of variable are the dependent variables. And the dependent variables, there can be more than one. That's possible to have more than one. But the dependent variables, these are the data that you are recording during the experiment. So these are the numbers that you are recording during the experiment. So a change in height, change in speed, change in weight. The last type of variable are the controlled variables. And the controlled variables are any other factors that can affect the recorded data, that can affect the dependent variable. And usually there are many different types of controlled variables that you have to keep in mind. And the important thing about the controlled variables is they need to be kept constant throughout the experiment for all the different groups. When you're setting up your experiment, you need to set up 2 different types of groups. The first group is called a control group. And this control group is going to provide a baseline for comparison. So this is going to show you what the normal results would be without the independent variable. So when you are setting up the control group, the independent variable is omitted or held at a normal level. So if the independent variable is something you can add, like adding fertilizer to a plant or adding vitamin to a diet, then the control group is the group of individuals who do not get the fertilizer or the vitamin, so you can see what the normal effect is. Some independent variables, like temperature or pH, cannot be removed from the situation. So when you have an independent variable that cannot be omitted, then it is held to the normal level. So for instance, for temperature, if you're looking at how different temperatures affect diffusion, you can't remove temperature, so your control group would be the group of subjects that are held at the normal temperature, which is often considered room temperature. The experimental group, this is a group where you actually have the independent variable, and often you have more than one experimental group. And for these different experimental groups, you have different levels of the independent variable. So often you will have 3 experimental groups, one with a low level of the independent variable, one with a medium level, and one with a high level. And so for instance, in this image, you have different plants and you are testing how the fertilizer affects the plants. This would be a plant in your independent group because it has 0 drops of fertilizer. Then you have 3 experimental groups. So this is your control group here. Control group, then you have an experimental group that has 2 drops of fertilizer, another one has 4 drops of fertilizer, and the third one has 6 drops of fertilizer. So your control group is giving you the normal baseline, how tall a plant would grow without fertilizer, and then you can compare the growth of these 3 different plants with 3 different amounts of fertilizer. The last part of the scientific method is to make a conclusion, and this is where you're determining if your hypothesis was valid or not, if it was true or not. And if it is valid, if your data supports the hypothesis, then the experiment can be repeated to make sure that that wasn't a fluke, that truly your hypothesis is correct. If the data does not support your hypothesis, then that is considered a new observation, and then you start over. So that was part of your observation, then you maybe refine your question and generate a new hypothesis, which you will then test with another experiment. An example of using the scientific method is listed in your lab report, and I'll just go through it here. So first stage is observation. So observation, maybe you have directly observed that your neighbor puts fertilizer on her tomato plants, and at the end of the summer she has lots of tomatoes. Maybe you have read in gardening magazines that it's important to add fertilizer, and maybe you have discussed with other people that the ones who use fertilizer, their plants tend to do better. So those are some observations you could have related to fertilizer and tomato plants. So the question you could form is, does fertilizer actually improve the number of tomatoes that are produced by a tomato plant? So your hypothesis could be, if I use fertilizer on my tomatoes, then I will have more tomatoes produced. So now it's time to determine your experiment, or to set up your experiment. So again, the three variables. First variable would be the independent variable. In this case, it would be the fertilizer, because you are controlling which plants get the fertilizer and which do not, and you're controlling the concentration of the fertilizer. Second is the dependent variable. Dependent variable, this is what you're measuring during the course of the experiment. So since you are focused on tomato yield, it's the number of tomatoes produced by each plant. So again, the dependent variable is the data you are recording during the course of the experiment. The controlled variables, these are the variables, anything else besides fertilizer that can contribute to the number of tomatoes that are produced. So that could be the amount of water that you give to the tomato plants, could be the amount of sunlight, it could be the type of plant that you have, it could be the location, the type of dirt. So any other factor that could influence the number of tomatoes that are produced. And all those controlled variables have to be exactly the same for each group. So the groups. You need a control group, so one group of plants where they receive no fertilizer, and then you would have three experimental groups. So one group of plants would get a low concentration of fertilizer, one would get a medium amount, and the third would get a high amount. So the only thing different between the control and experimental groups is the amount of fertilizer they are getting. Otherwise, they get the same amount of light, same amount of water, and same type of plant. Once the experiment is done, so you would say it'd probably go on for a couple of months, and you have all the recorded data, then you make your conclusion as to whether the fertilizer actually had an effect on the tomato plant growth and production. Another example for using the scientific method could start with observations on how chicken soup relates to a cold. Probably you have heard that chicken soup is good when you're sick, good when you have a cold. Maybe you have read that, maybe you've experienced it, that when you had a cold, you ate chicken soup and you felt like you got over the cold more quickly. So those could be observations, pretty common thought. So the question would be, does chicken soup really shorten the length of the cold? So to test this, you would generate a hypothesis, if-then situation. So the hypothesis could be, if a person eats chicken soup every day, then the symptoms of the cold will be reduced. So to set up your experiment, again, keep in mind the variables. So the first variable would be the independent variable. And in this case, the independent variable is eating the chicken soup. So that's what you're controlling, whether the people eat the soup or not. The dependent variable would be different variables you measure to see the symptoms of the person's cold. So there could be several of these. You could ask the person about stuffiness, you could measure amount of mucus production, inflammation in the upper respiratory system, but the dependent variables, these are the symptoms of the cold that you're measuring during the course of the experiment. And then the last thing, the control variables, this is anything else that could affect the length of the cold or the symptoms of the cold. So that could be the other food they're eating, how much they're drinking, how much they're sleeping, their amount of stress. And with experiments involving humans, it could be things like gender, it could be age. So anything else that could affect the length or the symptoms of the cold. So then you would look at the groups, you would set up your groups. You would have a control and experimental group. Control group would be the people who have a cold and do not eat chicken soup, and the experimental groups, maybe you have one group where they have soup once a day, a second group has it twice a day, and a third group has it 3 times a day. And then you run your experiment, you record the data, how fast all those individuals got over their cold or the level of their symptoms, and you make your conclusion. The last thing I want to go over is theory. And a theory is something that is widely accepted. So a scientific theory is something that is widely accepted because it is supported by a large body of evidence. So lots of scientists over a long period of time have performed lots of experiments on a certain idea, and it has been shown to be true through all of that evidence, all of that data. And a theory is not just one simple hypothesis, but it explains and connects many different facts. And so in general, a scientific theory is accepted as true. And the most famous theory that I'm sure you've heard of is evolution. Evolution is the idea that all living things arose from one cell on Earth, and it explains how you get the diversity and how things are continuing to change over time. Another theory is called the germ theory of disease. This one you may not be familiar with, but this is the idea where different microbes cause different diseases. And again, a theory is well accepted, and in the scientific community, it is viewed as a fact.